Hi, I'm Alex Thompson. I'm going to be talking to you today about some network analysis we've been doing in Oxford looking at the CSF proteome in ALS. The background really is that we can use network analysis of high-dimensional data sets to look for coordinated changes within the protein network and understand disease mechanisms. We used a technique called weighted gene correlation network analysis to construct a protein network of healthy control CSF proteomic data. This comprised 776 proteins which were allocated into 9 subnetworks or modules, with several proteins not allocated to an individual module. We then performed gene ontology enrichment analysis to look at the biological relevance of these networks, and as you can see on the right, module 1, which is that very tightly connected red module, was enriched for intracellular proteins particularly involved in transcription and translation. Module 2 was enriched for immune system proteins, and Module 3 membrane proteins. There was also enrichment in some of the smaller modules, which is also shown below. We then performed differential correlation analysis to see where changes occurred in the protein network in ALS and Parkinson's disease. In ALS, we found there was enrichment of changes within Module 1 proteins, including several TDP43 interacting proteins, tubulin beta, EEF1A1, YBX1, and some stress granule proteins. In ALS, there was also enrichment of changes in Module 3 proteins, so membrane proteins. In Parkinson's disease, we didn't find any module enrichment. We also performed longitudinal analysis of the ALS samples, and in longitudinally decreasing proteins we found enrichment of the membrane proteins, and these were mostly involved in guidance and signaling. In summary, the CSF protein network alterations we occurred in ALS were predominantly focused on pathways that have been strongly implicated in ALS and associate with TDP43 through interactors, through the involvement of stress granule proteins. Interestingly, the longitudinal changes in ALS related more to axonal guidance proteins, but overall the fact that there was involvement of several different parts of the protein network, we think suggests that successful treatment of ALS may require targeting of multiple different pathways.